Hello guys, in the last video we have explained the condensation and the decondensation of chromosomes during interphase. In this video we'll be explaining the DNA replication mechanism. So the DNA replication necessitates the intervention of an enzymatic complex. The enzymatic complex comprises first a helicase, then a DNA polymerase, and a DNA ligase. Since the DNA molecule is a double-stranded helix, so we need to separate the two DNA strands in order to form new DNA strands which are complementary to the original strands. That's why we need a helicase. The helicase, it unzips or unwinds the DNA double helix. It breaks the hydrogen bonds that connect the nitrogenous bases of the complementary strands at different places on the DNA molecule called eyes of replication or origin of replication or replication bubble. So the eye of replication corresponds to the site where the DNA double helix is unzipped. Next, the DNA polymerase comes and adds free nucleotides to the original strands by respecting the complementarity of nitrogenous bases. It adds A facing T and C facing G. So the DNA polymerase copies each of the two separated DNA strands that serve as template for DNA replication. At the end of the replication, we need a third enzyme called DNA ligase. DNA ligase will glue everything back together. It zips the new strand to the original strand together. So in blue here we have the helicase unzipping the DNA double-stranded molecule. Then we have the DNA polymerase here in yellow, which reads the parental or the original strand and which adds nucleotides by complementarity of nitrogenous bases. And at the end we have the DNA ligase that will ligate the newly synthesized strand to the original strand. Regarding the direction of replication, since DNA polymerase always reads the DNA strand in the 3', 5' direction, which means in the same direction as the opening of the double helix. So here we have the two anti-parallel DNA original strands. One runs in the 5', 3' direction, while the other runs in 3', 5' direction. The DNA polymerase can only attach to the free 3' ending of the DNA strand in order to synthesize the new strand in the 5', 3' direction. So, according to that, this makes it necessary for the two strands, which are also anti-parallel to their templates, to be made in slightly different ways. So we have two ways for DNA replication. On the first parental strand, which runs in 3', 5' direction, the strand below here, the replication is said to be continuous. It is done in the same direction as the opening of the double helix in order to synthesize the new strand in 5', 3' direction. So remember that DNA polymerase can only add nucleotides to the free 3' end of the DNA strand. However, on the other parental strand, which runs in the 5', 3' direction, the replication is said to be discontinuous since it takes place in the opposite direction from the right to the left here to that of the opening of the double helix. So, in this way, the new strand can only be synthesized as a series of fragments, small fragments, that will then be glued together by the DNA ligase. At the end of the DNA replication, the two new DNA strands formed are called the newly formed strands or the newly synthesized strands. So, I repeat, since the two original DNA strands are anti-parallel, running in different directions, the DNA replication takes place in different ways, in opposite ways on both strands. On one strand, the replication is continuous. It is done in the same direction as the opening of the double helix, while on the other strand, the replication is discontinuous. The DNA polymerase synthesizes small fragments facing the original strand in order to be glued together, zipped together by DNA ligase. So here a video explaining the DNA replication during interphase. So the DNA replication copies the genetic information of a cell. Now, why must DNA replicate? First, each cell needs a complete set of DNA in order to be functional, to make proteins, to divide, etc. And before a cell can divide to make more cells, it has to replicate its DNA. So let's look to the steps of DNA replication. The first step, we have a DNA molecule that has two original strands because remember we have a double helix, so two strands wrapped around each other. So how do we copy that? We need a couple of enzymes to help the cell performing DNA replication. So enzymes like first helicase. Helicase is going to actually unzip the DNA molecule because remember the two DNA strands are connected by hydrogen bonds, and helicase is going to break those hydrogen bonds between the nitrogenous bases, and therefore we will have two unzipped separated DNA strands, which are the original DNA strands. So helicase unzips at different location called origin of replication as shown in this figure. In the next step, we have another enzyme called DNA polymerase that is going to add free nucleotides that pair up with the original strands. A pairs with T, C pairs with G. So here the DNA polymerase comes in and starts adding nucleotides to each of those DNA original strands because we want to make a copy. We are going to end up with two double helices at the end of DNA replication. So the DNA polymerase adds free nucleotides to the original DNA strands. And in the last step, after copying the DNA, we need a DNA ligase that will glue everything back together. It zips the new strand we just made to the old original strand. Thus, at the end of DNA replication, we obtain two identical DNA molecules, each one consisting of an original DNA strand and a newly synthesized strand. So each of the two DNA molecules obtained contains an original strand and a newly synthesized one. So Because of this, we call the DNA replication a semi-conservative process because, as you can see in the figure, in blue, we have the two DNA original strands. Now, in the two DNA molecules obtained, each of the two DNA molecules obtained contains an original blue strand here and a newly synthesized one in red. So this process is called semi-conservative because each DNA molecule will contain an original strand and a new one. Remember, the semi-conservative process, we are conserving a part of the original DNA molecule, the original strand in blue here. At anaphase of mitosis, the sister chromatids, which are formed during DNA replication of each chromosome, separate, and each one moves into a daughter cell. The sister chromatids contain the same genetic information. They are identical. So DNA replication is necessary in order to conserve the genetic information that is transmitted from the mother cell to the daughter cells after cell division. Here a simple diagram that shows the conservation of the genetic information and the semi-conservative process of DNA replication. At the beginning of interphase, the chromosomes are made up of one chromatid. I have represented here only two homologous chromosomes, only one pair. The two homologous chromosomes are represented in different colors, in blue, in green, and the vertical lines correspond to the DNA strands. Each chromosome is made up of one chromatid, which means one DNA molecule, two original strands. After DNA replication, at the end of interphase, the chromosomes are now made up of two sister chromatids. The red lines and the yellow lines here correspond to the newly synthesized DNA strands. So we obtain at the end of interphase chromosomes with two chromatids united by a centromere, the black spot here. At the end of mitosis, we obtain two daughter cells. Each of the two daughter cells contains the same number of chromosomes as the mother cell. The daughter cells are identical to each other and to the mother cell. At the end of mitosis, the chromosomes are made up of one chromatid because sister chromatids separate during the anaphase of mitosis. So at telophase and cytokinesis, the chromosomes are made up of only one chromatid, and each chromatid contains an original strand and a newly synthesized one. An original strand and a newly synthesized one. So both cells are identical to each other's and to the mother cell. That's why we can say that DNA replication and mitosis allow the conservation of the genetic information. Thanks for listening.